So, in all, this is the energy that is liberated, or that is the useful energy, useful energy. In same terminology, we can use this format, delta E 0 prime. What is this? Or this is useful energy. For knowing this, we must know what exactly this is. This notion is called as standard redox potential. Now, what is redox potential? Redox potential is the ease with which any substance donate or accepts the electron. It is the capacity of a substance or substrate to donate or accept electron, or in a better word, it is fugacity with which electron escapes the substance. Okay. Fugitive. What do you mean by fugitive? Person on run away. Okay. So whenever electrons they run away from one substance and been accepted by another. So this capacity of any substance to donate the electron and accept the electron is called as redox potential. Reduction oxidation potential. Okay. Reduction oxidation potential. This, whenever it is standardized, is called as standard redox potential. That is E0 prime. You know the standard condition is 1 mole of substance, then pH 7 at 37 degrees centigrade. So electrons they are transferred from one substance of having higher redox potential to donate electron to a substance which accept it, and the difference between the redox potential of two substances is shown by delta E, or just delta E. So that is the difference. Now why we should know this? Because we want to know how much energy is liberated while transferring the electron from the donor to the acceptor, and it is given by one formula which says that the energy liberated can be given by NF delta E. N is number of electrons, F is Faraday's constant is around 23 kilocalories per volt, and this is the difference of redox potential. Remember this. Okay. The energy that is liberated. Why we want to know the energy? Because that energy is used for the capturing in the form of ATP. How much energy is liberated, whether that is useful to do work, which work is done, and how it is captured, for the formation of ATP. So we know that this energy that is liberated, it depends on, because and we know that 1 or 2 electrons they are transported, this is constant. So whatever difference in the redox potential is there, that is the main determinant of the energy that is useful. Energy that is useful. Now there are certain substances which can donate electrons. They are having reducing power. They themselves get oxidized. For example, hydrogen. Okay. Then comes NAD or NADH. So this is redox couple. Okay. So whenever it get reduced, it is H, NADH. Whenever it is oxidized, it is NAD. So redox potential of this particular substance, because we know that while oxidizing various substrates in our body, in glycolysis or TCA cycle, or in case of the fatty oxidation, beta oxidation of fatty acid, there is formation of NADH. Okay. So this has got around minus 0.032 volts of redox potential. And last is O2H2O, which has got plus 0.82 volts. Why I am keeping, I will give the names or the substances in between. So first, foremost, this donates electron, oxygen, it accepts electron, get converted into water. Oxygen is reduced to water. So this oxygen has got highest accepting policy of electron. It is used to accept the electrons. NADH, it is used to donate electron. And there are certain substances in between, like FAD, okay, coenzyme Q, or it is also called as ubiquinone. Then cytochromes, cytochrome B2 AA3. So this is around minus 0.2. Cytochrome C is around 0.08. Cytochrome B likewise 0.1 or 0.12, something like that, volts. So this is again, from here it becomes positive. This indicates, sign indicates that they are having highest potential to give up electron. This sign, plus sign, shows that they are highest to accept the electron. So electron flows like this. So there is no direct transfer of electron. So you can see that it is a stepwise, one after another. So these substrates or these things, these are the enzymes and coenzymes. We will see it later on. They are arranged in a increasing redox potential. It is said that it is increasing because when we start from, this is 0, this is minus, this is plus. This is we call as increasing redox potential. So they donate electron, here they accept electron. So this is increasing redox potential. So these particular substrates, they are arranged in the increased redox potential. So these are the substrates. Now what is the use of this? Whenever you want to see how much work is done, we will come to this again, or how much energy is liberated when electrons, they are passed from minus 0.32 to plus 0.82 volts. You can have its delta E0 prime. Okay. So depending upon the redox potentials, the redox substances or substrates, they are arranged. Now how they are arranged, whether they are lonely, why they are arranged, what is their role, we will see their role in the transfer of electron and capturing in the form of ATPs. Okay. So electrons, they are transferred, or they are coming from one substrate to another substrate. This is called as oxidation, as we know, and this has been linked with the formation or synthesis of ATP. So this is oxidative phosphorylation. Okay? Now before going to that, we will just see regarding a favorite structure called as mitochondria. Okay, this is mitochondria. This has got outer and inner mitochondrial membrane. OMM. Okay. Then this is intermembrane space. And this is matrix. So you know the outer membrane. This is in 2 dimensions. In 3 dimensions, you can just see elongate structure of a mitochondria. Okay. This is not simple planar structure. Okay. You can imagine that this is a space occupying 3D structure of. So outer one is outer membrane, which is permeable for most substances. Inner membrane is not. Okay, not permeable. It do not allow all the substrates or substances to enter inside. So it requires another system that is called as shuttle system. We will see later on. So that the substances which are synthesized inside the mitochondria in matrix to be transported out, and some of the substances which are there out of mitochondria in the cytosol, they have to be traveled to the matrix. Okay? So this is mitochondria. The inner membrane of the mitochondria is having folds, multiple folds. It is folded. Why? To increase the surface area, and on these folds you get some structures over it. And these structures, they are nothing but the electron transport chain. These structures are nothing but electron transport chain, and whatever we have seen previously, these substrates, they are arranged in this electron transport chain in their increasing redox potential. So the -1 is on the left side and plus is on the right side. Means the electrons, they are transferred from the NADH fired into the OP. Now these substrates or enzymes and coenzymes, they are not solitarily present in the mitochondria. They form the complexes, and they are seen as these structures over the inner mitochondrial membrane, and these are called as complex 1, 2, 3, 4 and complex 5. So all the substrates, enzymes, coenzymes required for the electron transport, they are arranged on the inner mitochondrial membrane in a lipid protein complex form. So this is complex. Complex means it constitutes various subunits in it. So there are various proteins, coenzymes. They form a complex, and that is embedded inside the inner mitochondrial membrane. Okay, and this inner mitochondrial membrane, forms a complex, and this complex are nothing but lipid protein complex. You know that inner mitochondrial membrane is having highest quantity or concentration of cardiolipin, and sometimes it is said that inner mitochondrial is a special membrane which is not equivalent to the cell membrane, or which may not conform to the fluid mosaic model of the cell membrane. So it has got slightly different constituents, it has got slightly different mechanism and properties. So inner mitochondrial membrane is a different thing for our understanding. So these complexes, how they are arranged in a increased redox potential. How they are arranged? We just magnify this portion over here. Okay. So what we are looking at is a electron. What we are looking at is a electron transport chain. So this is inner mitochondrial membrane. I am just magnifying it. This is inner mitochondrial membrane. Outer mitochondrial membrane is somewhere here. Okay. This is outer. This is inner. This is space. And here is a matrix. What is matrix? This contains the enzymes, coenzymes for tricarboxylic acid cycle, beta oxidation, and other oxidative processes. The enzymes and coenzymes required for these cycles or reactions, they are there in the matrix. So matrix is the final destination where final metabolic pathway it occurs. Okay. Does matrix revolution? Do you know what is matrix? Matrix revolution. Do you know Keanu Reeves? So is it related somewhere? Okay. So that is another sci-fi movie of Keanu Reeves, and 2-3 parts are there of matrix. So this is not related to this matrix. This matrix is mitochondrial matrix. Okay. So it contains enzymes and coenzymes for the TCA cycle and beta oxidation. These are the major catabolic pathway where the electrons are transferred from the substrates to NAD, or they get substrates, they get oxidized in the TCA cycle or beta oxidation. So how they are arranged we are now going to see. First is called as complex 1. Okay. Complex 1 is L shaped or it is boot shaped. This is complex 1. Whether boot comes first or L comes first, I don't know. But this is what it is said, that it is L shaped or it is boot shaped. This is complex 1. What are the constituents of this complex 1? This is called as NAD dehydrogenase. This complex is called as NAD dehydrogenase or NADH dehydrogenase or NADH CoQ oxidoreductase. Complex 1. It contains this enzyme. Okay. Then it contains FMN, that is flavin mononucleotide, and it contains iron sulfur centers. This is another special component of electron transport chain. I have not given this before because it is pertinent to tell over here only. This is called as iron sulfur center. This is non-heme iron. This is non-heme iron. What are heme irons? Hemoglobin is heme iron. Then cytochromes, then catalase. So there are number of examples of heme iron where heme is present and within the heme there is presence of iron. But in case of iron sulfur centers or iron sulfur clusters, these are the proteins where heme is not present. Okay. And they may be present in FES format or Fe2S2 or Fe4S4. So this is Fe. These are the sulfurs, and these sulfurs are from cysteine residues. So this is Fe1 center. Okay. Then there are Fe. These are two inorganic sulfurs. They can be joined to two Fe's. And these two Fe's then can be joined to the cysteines again, similarly on that side of protein. Okay. Then other iron sulfur is they are alternate. This is Fe, this is S, this is Fe, this is S, this is S, this is Fe, likewise. So 4Fe, 4S or Fe, 4S4, and this is linked. Fe is linked to the cysteine of protein. It is a protein that is present in complex 1. Okay? It is present in complex 1. So complex 1 is made up of various subunits. Important subunits are FML, enzyme and iron sulfur centers. These are the constituents of complex 1. Complex 1 is having around 46 units in it. Total number of units in complex 1 is 46, various enzymes, coenzymes or subunits, out of which 7 are coded by mitochondrial DNA. This is another idea that the electron transport chain, or for that matter many of the mitochondrial proteins, they are synthesized from mitochondrial DNA. So each cell it has got two DNAs. One is nuclear DNA, another one is mitochondrial DNA. So in mitochondria you have got this DNA. This is circular DNA, two-stranded circular DNA, and as having two chains. One is called as H or heavy chain, another is called as light or L chain. L and H, these are two chains which are present in mitochondrial DNA. Now this gives, or this throws light that a single cell is having two different genomes. One is mitochondrial, another is nuclear. So the mitochondrial proteins, they are synthesized from mitochondrial DNA as well as from the nuclear DNA. And it is said that at the start of life, the aerobic organisms, okay, they form a symbiotic relationship with the bacteria, oxidizing bacteria. Okay, and this symbiosis is still going on there, and these bacteria, they are converted into mitochondria. So each of our cell is having a number of bacteria in it, and these bacteria, they are helpful, and without that bacteria life is not possible. Same is true for plant also, where mitochondria are there, but for photosynthesis, cyanobacteria they form, or they have a symbiotic relationship with plants. So what cyanobacterium is converted into? It is converted into chlorophyll, thylakoids. Chlorophyll, that is nothing but a cyanobacteria and oxidizing bacteria. They are present in eukaryotes. So in eukaryotes, the mitochondria are nothing but the symbiotic bacteria. Somewhere in the evolution of life, we have symbiotic relationship with the bacteria, and with that bacteria, for that matter, life is possible due to that. So it is a reality that life depends on two bacteria: cyanobacteria, which uses energy for splitting of using carbon dioxide, oxygen for the formation of, it evolves oxygen, it uses carbon dioxide and sun energy for the formation of carbohydrates. And another bacteria which takes these carbohydrates and split it and gives out oxygen again. Okay. So carbon dioxide is not directly evolved in the respiration. Carbon dioxide is there evolved in case of TCA cycle. It is not through this oxidation reduction process. Okay. So two bacteria are there. One is cyanobacteria, which is important for the synthesis of a substrate, and oxidative bacteria, which splits this carbohydrate, gives up oxygen, used oxygen for the formation of water. Okay, and carbon dioxide is given. That carbon dioxide is again used by the cyanobacteria for the synthesis of carbohydrates. Oxygen is evolved. This carbohydrate and oxygen again is taken up by the oxidative bacteria, get converted into energy, and again carbon dioxide is liberated. So this cycle goes on. So this is very important to know. Why it is important to know that mitochondria is different DNA? Because there are certain inborn errors or certain diseases. This is called as mitochondrial diseases or Oxford diseases. Okay? For that, for so many years, it was not possible to pinpoint the mutation. It is seen that certain diseases, they run in family, so there is inheritance. But nobody looks to the mitochondria. They always look for the nuclear DNA. What is wrong with nuclear DNA? There is nothing wrong in nuclear DNA. There is wrong or some mutation in the mitochondria. It is just in last 25-30 years this is evolved, and this particular thing is mitochondrial diseases, and mitochondrial medicine is a separate branch of medicine. Very interesting and evolving branch of mitochondrial genetics and mitochondrial diseases. So whenever we come to know that some of the components, they are synthesized from the mitochondrial DNA. So that's why it is very important to know mitochondrial DNA, its structure and its mutation. Okay? So what happens in this? What exactly is done with the happens with complex one? That energy or reducing equivalents which comes from the substrate, glucose for example, it is converted into lactate or pyruvate, whatever. So NADH is given. This NADH donates its protons to this complex one, or complex one snatches it and it is get converted to NAD, which can be again used for the oxidative processes. This FMN is converted into FMNH2. It is converted into FMNH2. Iron sulfur centers, they also help in the electron transport. Now, while transporting, there is drop in the redox potential from NADH to complex 1, which is FMN. There is slight drop in the redox potential. Now this is used to do work. Which type of work? Due to this, there occurs conformational change in the complex 1, and this complex 1, it pumps 2 electrons in the inner mitochondrial, 4 electrons in the inner mitochondrial space. So there occurs pumping. Here, electron or reducing equivalents, they flow, hydrogen, hydride or electron. Similarly, at the same time, from the matrix side, 4 protons, they are pumped to the inner membrane space. Now, there is another component called as coenzyme Q, or it is also known as ubiquinone. Coenzyme CoQ or ubiquinone. This is lipid soluble. It contains isoprenoid units. Isoprenoid unit is nothing but having 5 carbon atoms with it. So this is lipid soluble, or it can move. Rather than soluble, we can say that it can move from its original position to any direction. So it accepts the reducing equivalents from this complex and is converted into reduced phenol. So this is complex 1, where the reducing equivalents, they are transferred to the ubiquinone. Ubiquinone is nothing but is a lipid carrier, or this is a lipid which contains isoprenoid in it, and due to that it can move across and vertically and horizontally in the inner mitochondrial space. It carries the reducing equivalents and get converted into QH2, reduced phenol. This is phenol, this is quinone. So it can be converted intermediately to semiquinone, where it accepts 1 hydrogen and 1 electron. So that is denoted as, and then converted into QH2. Okay? So this is how it occurs. So it is present in the form of ubiquinone, semiquinone and ubiquinol. Oxidized, fully reduced and half reduced. These are the forms of ubiquinol. So this is complex 1. In between there is complex 2. I can show here complex 2. Okay. This complex 2 is called as succinate dehydrogenase. This is complex 1. This is complex 2, which is succinate dehydrogenase or succinate CoQ oxidoreductase. This contains succinate dehydrogenase enzyme. Okay. It also has got FAD in it. And again, iron sulfur centers. This complex 2, it is totally made from nuclear DNA. This is totally made from nuclear DNA. What is the role? Same, that from substrate like glycerophosphate or from acyl CoA, that is fatty acid, FAD to complex 2, and from complex 2 the reducing equivalents, they are given to again coenzyme Q and converted into QH2. Now the potential drop or the energy liberated is not used, or there is no pumping of protons from complex 2. Okay. There is no pumping of protons from complex 2. Then comes, so ultimately both complex 1 and complex 2, they donate a reducing equivalents to the Q to form QH2. Here in between there are electron transfer pleoproteins. They are seen with glycerophosphate. dehydrogenase, okay, which then transferred electron to the complex 2. So ultimately what we get is QH2. Now here onwards electrons are transferred as reducing equivalents rather than H or hydride. It is electron that is transported here onwards, and there is presence of another complex called as complex 3. Complex 3 is called as Q cytochrome C oxidoreductase. Or cytochrome C reductase. It contains two cytochromes. Actually, B and C are two cytochromes. There are two Bs, BH and BL, and one cytochrome C1. These are the cytochromes which are present in the complex 3. It has also got iron sulfur center. Now, the iron sulfur center which is present in complex 1 and 2 and complex 3, they are slightly different. In complex 3, the iron sulfur center present is called as iron sulfur center of risk or risk. What is the difference? The iron in case of, or sulfur in case of complex 1, complex 2, they are linked to the protein through cysteine, but here it is linked through histidine. Iron sulfur centers or the proteins which has got this iron in it, sulfurs in it, they are linked with the histidine. That's why the name is risket, iron sulfur centers. Now what happens here? Again the QH2 is converted into Q. Okay. It donates its electron to this complex 3. Okay. Complex 3 also pumps 4 protons while transferring the electrons. Now here electrons, they are transferred from one QH2 to finally the acceptor. We will see what acceptor is. It has to travel through QH2 to BH, BL, C1 iron sulfur centers, and finally iron sulfur centers C1, and finally to the acceptor. That acceptor is cytochrome C. And this is done with the help of called as Q cycle. You just remember Q cycle. That means, see, up to here we have got 2 electrons, 2 protons. Now here onwards, each cytochrome we have seen last time, it accepts 1 electron at a time. It cannot access. So in cytochrome, the iron, it has got 2 forms, or it is interconvertible. Iron can be converted into ferrous form, ferric form. When it accepts, it is ferrous form. When it donates, it gets converted into ferric form. So this occurs, and a single electron is required. So at a time, single electron is taken up by these cytochromes. Okay, one electron is given to this BH, BL form. Another electron is given to FeS4. From here it goes to C1, and from C1 to the cytochrome C. So these are the 2 ways. 1 electron is given to this BH, BL, then again from BL it is given to Q. From Q again it comes to FeS4. So that is called as Q cycle. If you remember it, it's okay, or just remember that Q cycle is important for the transport of electron from QH2, from ubiquinone to the complex 3. And finally from C1 it is given to the cytochrome. So Q cycle, it occurs inside the complex 3. Okay. So QH2 is converted to Q is converted to semiquinone, semiquinone again converted to QH2, and one electron is given to BH, Bl, then it is again given to Q, first Q. So there are two systems going on and converted to QH2. Another is given to the iron sulfur centers, FeS, another electron, and from FeS it is given to C1 to cytochrome C. If you remember, it's okay. So this is the function of complex 3. So it has got various components in it. Okay. So it has got one component from mitochondria out of 11 components. Total components are 11, out of which one is from mitochondrial DNA. One component is formed. Now from complex 3, it is given to the cytochrome. From complex 3, electrons are given to cytochrome C, and cytochrome C is the one component of electron transport chain where the electrons can be taken up from this side and it is given to next complex. So that's why cytochrome C is called as a mobile carrier. It is mobile, it can move in a horizontal direction. Okay. So these are complex 1, complex 2, complex 3, and lastly there is complex 4. This complex 4 is popularly known as cytochrome oxidase. This complex 4 is known as cytochrome oxidase. Now what is oxidase? We have seen last time. What is oxidase? Oxidase is the enzyme which uses oxygen as hydrogen acceptor. So same thing is done over here. This electron that is carried with the help of cytochrome C, it is given to complex 4. Now complex 4 has got multiple units in it. These are copper center A, copper center B, then A and A3, or commonly it is called as AA3. This is heme. So it has got copper center A, copper center B, and AA3. These are the heme. Cytochrome oxidase is the enzyme. Okay. So electrons first they are taken up by cytochrome B. This is what happens. Sorry, copper center B, it collects electron, then given it to the A, cytochrome A to cytochrome A3 to copper center A. This is how electrons they are transported in this complex number 4. Okay, and finally the electron is given to the oxygen. Here electron comes and some of the hydrogen which are there in a matrix. So electrons, oxygen and matrix proton, they form water. This is the final product of the oxidation. While transferring this, there is again pumping of 4 hydrogens across the mitochondrial membrane. So 4 hydrogens they are transported across mitochondrial membrane. Are you getting me? This is very simplified form of electron transport chain. Okay, very simplified form. Now it contains 3 mitochondrial DNA components out of 13. Around 3 are from mitochondrial DNA. So the proteins of complex 4, 3 proteins they are from, or they are transcribed and translated from mitochondrial DNA. Okay, so this is electron transport chain proper. Now what happens next? What happens next is this transported protons in the inner mitochondrial membrane, they get accumulated. है ना? So relatively more protons or more hydrogens are in the inner mitochondrial membrane space, internal membrane space, than in the matrix. Whenever there is flow of electron from substrate finally to the oxygen. So what happens in this electron transport chain? First, what is electron transport chain? It is the arrangement of all the substrates or substances in their increasing redox potential in the form of complexes in the inner mitochondrial membrane, so as to transfer or transport electron in a stepwise manner finally to form water. Oxygen is reduced to water. So this is the electron transport chain and its assembly. This is organization of various components of electron transport chain in 4 complexes. There is another complex called as complex 5, which is called as ATP synthase. We will see this ATP synthase in next lecture. So this is about the organization of electron transport chain. Okay. Now again come to a first thing first, that is delta G. How much energy is liberated from N? There are 2 electrons that are transported into 23, that is Faraday's constant, into, yes, what is the drop of redox potential? What is delta E? Delta E 0 is equal to, first is NAD is having minus 0.32 minus plus 0.82. कितनी येता है? How much it is? Minus 1.1 volts is the drop of redox potential through which electrons they are transported. So this comes about 46 into, sorry, this comes about minus 46 into 1.14. Now this is up to you to calculate this, how much energy is liberated, and keeping in mind that 1 ATP when it is hydrolyzed gives 7.3 kilocalories. It means that for the synthesis of ATP you require 7.3. It is around 46 into 1, 46 plus 5 something, 68 च्या जवळपास काहीतरी येतं. Okay, you just calculate if that is the energy that is liberated, delta G is the energy that is liberated, how many ATPs will be synthesized, and we'll see how many actual ATPs are synthesized in a process called as oxidative phosphorylation. Okay? So energy liberated is 46 into 1.14 volts. this is calories. This is what totally delta G is kilocalories. So 46 into 1.14 is the kilocalorie is the energy that is useful to do work. So how much is captured in the form of ATP and how much is used to pump protons and how much is wasted as heat. We will see in next lecture. Any problem? Any difficulty? Yes, questions? Okay. Now these sessions of oxidative to phosphorylation, they are quite out of interest of many of us, whether he is from faculty side or student side. This is kept as an option, or you just mug up the things regarding electron transport chain, but there is a vast evolution regarding mitochondria, because mitochondria are not only important for this, or they are not only the energy house, but they are also implicated in various diseases till that we don't know, like Alzheimer's, like cancers, various cancers, then Parkinson's, and then mitochondrial diseases, and aging also. The process of aging, it also depends on mitochondrial dysfunctioning. So that's why we must know the details of mitochondria, its functioning, and mitochondrial genome. So that's why this is again we are taking live on YouTube. So whenever time permits, you can watch it again and again. Any difficulty? Okay, thank you.